Hi everyone. I'm really delighted to participate in this wonderful symposium that seemed long overdue somehow, but I think it's a really exciting initiative, and good luck with the continuation of this initiative on the woman brain. So my lab is interested in the neuronal circuit that drive social behavior, and we would like to understand social behavior in mechanistic terms, which means try to understand the specific neuronal population within the brain that organize distinct social behaviors. And as a young investigator, when I started my lab as a woman investigator, I say, well, unlike many other labs, I will make sure that we study both male behavior and female behavior in parallel. It has proven challenging in some way, but also I think extraordinarily revealing. And what we are trying to understand is really the whole circuit that lead to behavior, from the detection of specific stimuli in the outside world, how these stimuli are processed by sensory areas of the brain, and how in turn these cues and the processing of these cues according to the internal state of the animal lead to distinct social behaviors and interaction between males, between females, between male and female and their young. And one really interesting aspect of social behavior is their sexual dimorphism in nature. It's very clear that the same stimulus detected by a male or by a female will lead to extraordinarily distinct behavioral outcome. So what generates these different outcomes? Are there specific neural circuits that exist in a male brain and not in a female brain that lead to these sex differences? Or in other words, where is the sexual dimorphism observed in behavior originating from? And when we start to study this entire circuit from the sensory stimuli to the behavioral outcome, we came across some really interesting findings. One is that obviously, and this has been obviously very described in the literature, the signals that lead to specific social behavior outcome are extremely species specific. And it makes sense. If you're a bird, you want to be able to detect a bird of your own species. And if you're a mouse, you want to detect another mouse, and the cues are extremely different. So for example, in this American flicker, in this species, the way male and female distinguish each other or determine that they are males or female is the presence of this little black spot here in the face of a male. So if you mask this black spot, so you paint over this black spot, now the other male are going to attempt to mate with this male without a black spot because they think it's a female. And similarly, if you paint a black spot on the face of this female, the other male are going to attack this female because they think it's a male. So I have a black spot, I have a mustache, I'm a male, I don't have a mustache, I'm a female. So that's what the bird behavior in this particular species relies upon. And in mice, we discovered that pheromones are absolutely essential for sex discrimination. And if you use a mutant mouse that is unable to detect pheromones because it has a non-functional voronasal organ, then males are unable to tell apart a male from a female and will mate indiscriminately with males and females. Moreover, if you now look at a female that has a non-functional voronasal organ, that female has an extraordinary strange behavior. It now starts to behave like a male and will attempt to copulate with other males and females as if it was a male, suggesting that the circuit underlying male and female behavior exist in both the male and the female brain, but are regulated in sex-specific manner. And obviously, the question is, how would that work in human? And obviously, I'm not going to talk about this. But what I will tell you about today is a story on a particular behavior, parental behavior, that is often considered as a female-specific behavior, and how this circuit is organized. And interestingly, the central circuit of social behaviors are thought to be evolutionarily conserved across vertebrates or mammals, and so this is an aspect that is obviously of high interest. So why parenting? What about parenting? Well, parenting behavior is quite an unusual social behavior. We all take it for granted. Obviously, we all benefited from parenting, and some in the room are themselves parents. We take it as a very obvious behavior. But if you think about it, it's a very unusual behavior. First, why would an animal take care of this helpless individual that screams a lot, that require a lot of energy, resource, generate a lot of sleepless night? For human, we understand the social context, right? We obviously need to take care of our young, but for a mouse or a rat or an animal, why would it take care of that helpless screaming thing? And what that means is that the circuit that underlie parenting behavior are to a large extent genetically pre-programmed, such that this circuit now drive the animal towards caring of the young. And what is interesting about parenting is that it's a long-lasting social behavior. In human, particularly long. It involves years of care, probably lifetime care, also a lot of sacrifices. Parenting in human is interesting because it involves more than strictly the parents. It involves the extended family, the friends, and even the entire social structure, forming this very unusual alloparenting that is found in relatively few species. And then finally, parenting obviously is essential for both the emotional and physical care of offspring. So this is the rosy picture of parenting, but parenting quite often actually goes wrong or doesn't go as well as one would imagine. And in very high frequency, one encounter a patient with postpartum depression, which affect 20% of mothers, 10% of fathers. And frankly, I do not know of a mental illness that affects such a large population, such a large proportion of people. And the symptom is difficulty in emotional connection and caring of the offspring. It is accompanied by extreme worry, dread, feeling of failure, and lead to difficulty in bonding with the child and obsessive thought of harming a child, although in the case of postpartum depression there is no action, which is different from a more rare form of postpartum illness, postpartum psychosis, that affect a very small minority of new mother, but lead to this obsessive thought that might lead to action and therefore require hospitalization. I will read now a quote from a patient with postpartum depression to make this poignant call very, very vivid. Sometimes I get really down and feel like, man, I can't do this. No one talks about the low moments, the pressure you feel, the incredible letdown every time you hear the baby cry. I've broken down I don't know how many times, or I'll get angry about the crying, then sad about being angry, and then guilty, like, why do I feel so sad when I have a beautiful baby? The emotions are insane. You can really, I think, vividly feel the anguish of this mother. And the reason I'm putting here this particular quote is because of the person who said this, which is Serena Williams, who for many women epitomizes women's strength. And so, this is an illness that can affect anyone and lead to this very emotional roller coaster. So there's something really interesting about parenting and the regulation of parenting and parenting in both health and illness. So obviously we're not studying human, we're studying animals. And in animals, in mammals in particular, there's a very interesting sexual dimorphism in parenting. At least in most mammalian species, the female is the primary caregiver, and it makes a lot of sense because through pregnancy and then lactation, females invest enormously in the birth and the caring of the infant, so it makes sense that they are the primary caregiver. In about 60% of the mammalian species, males have a very different behavior towards offspring, towards infant. They kill them. And this infanticidal behavior was actually discovered here at Harvard in the late 70s by Sarah Hardy, who was looking at this particular species of primate, the langurs, and observing that each time a male would enter a foreign social group, then all the infants would disappear. And what was interesting that is her PhD, by the way, what is interesting in her her thesis is that not only she makes this observation, but she provides some evolutionary explanation for this, which is that a foreign male has no chance to sire their own offspring unless they kill the offspring of their rivals, and now the females are becoming sexually receptive. So now they can sire their own offspring until another male will come and kick them out, etc. So the idea is that this infant-mediated aggression is a common evolutionary-driven behavior in males. It's observed in about 60% of the species. Not all the species develop infanticide, and they're thought to be evolutionary response to infanticide. One of them is monogamy. If the male and the female stay together, there is no need for infanticide. Dilution of paternity, in which the female mate with multiple male, and therefore none of them know which is their offspring or not, and therefore do not kill them, etc., might be adaptation against infanticide. And then finally, female can also neglect or attack infant, particularly in situation of stress. So this has been observed in the wild or in our mouse or rat colonies when there's a lot of noise, a lot of stress, when in the wild there are a lot of predators or there are not enough food, females tend to kill their own offspring in order to wait for better circumstances to make sure that her offspring have a chance to thrive. If they have no chance to thrive, it's just no need for them to waste resources. So this behavior, therefore, is both observed in males and in females. So we came into the observation of parental behavior because when we started, we really thought about parenting as a female-specific behavior in mice. So in mice, there's a very clear sex difference. If you take a virgin female and put her with pups, she will immediately build a nest and care of the pups. So she's spontaneously parental. If you take a virgin male, the virgin male are spontaneously infanticidal. They will immediately attack the pups. And we were extremely interested in this sex difference. As I told you in the introduction, when we looked at voronasal mutant female, they behave partially like males. And so we thought, well, let's look at voronasal mutant male and see whether they partially behave like females. And so this female behavior that we tested was parenting. And indeed, virgin male that are deficient for the voronasal organ no longer attack infant and in fact are parental. So that satisfied our hypothesis about the voronasal organ controlling circuit of male and female specific behaviors. But then we thought, why would the brain have male specific behavior or female specific behavior circuit in the brain of the other sex? What's the reason for this? And in the case of parenting, there was something very interesting that we found in the literature, which is that males, after they mate with a female, not immediately, 3 weeks after they mate with a female, actually switch their behavior from infanticidal behavior to parenting. 3 weeks is exactly the gestation time, and so it makes sense that a pup emerging 3 weeks after mating would be the male offspring, and so he better not be infanticidal. And so to show you this amazing behavior, this is a male that 3 weeks earlier was infanticidal, so this is certified infanticidal male. We make that male mate with a female, and 3 weeks later you see that this male dutifully retrieved these pups. They are not even his own offspring. He will take care of any pup 3 weeks after mating. So he retrieved the pups and, good dad, now try to make sure nobody is left behind. And then going back to the nest and then huddling with the pup. So you can see this extraordinary switch in behavior that clearly show that indeed the male brain contains fully functional parental circuit. Now, at the evolutionary level, this is actually very interesting because if you look in nature, we all think about parenting as being a property of mammals, but actually there are insects that are parenting, fish, here frogs, reptile, birds, and mammals. Here, this is a female taking care of her little bugs. This is a fish that has larvae in his mouth. This frog, as you can see here, this is a female frog, had a tadpole here that she protects and will bring to a source of water. But very similar species have variation in which both parents are taking care of the pups, or sometimes even the male taking care of the pups. And so I like particularly this example here that was highly studied by my colleague Lauren O'Connell, who is now a faculty at Stanford. You see here a female with a tadpole, and in these two species the male also participate in protecting the tadpole. You can see the tadpole right here. And so what this indicates is that evolutionary speaking, this male and female circuit are actually both in the male and the female brain, and it makes sense because it gives a lot of flexibility to evolution, such that species that are genetically similar can adopt some behavior or not according to the sex and according to the species. So in this case, only the male use parenting circuit, here both male and female, and here only the female. So we think, this is a really coherent picture at the evolutionary level. So what about the mouse? Well, in mice, males have this little harem, and so there's a dominant male with a bunch of females. They have communal nests, so they take care of the pups in turn. And so these males mate with the females, so they are parental. But subordinate males that do not have a chance to mate are infanticidal until they kick out the dominant male, kill the pups, mate with the female, and then become parental in turn. And so from a neuroscience perspective, this is quite fascinating because it indicates that there are in the brain two types of circuits, the one driving infanticidal behavior and parenting behavior, and according to the state and the sex of the animal, one circuit or the other will be used. And so what are these circuits and how are they regulated? Several years ago, a graduate student in my lab used a very simple technology, which is the induction of immediate early gene CFOS upon neuronal stimulation to look at areas that were activated in the brain of males and females when they were parenting. So the idea is to have a male or a female with pups, observe their behavior, sacrifice them half an hour later, and then look for areas in which there was a lot of CFOS activation after parenting. And what we found is that in the preoptic area, there was a very high induction of CFOS in parental males and parental females. Now, this is absolutely not surprising. The finding of the involvement of the preoptic area in maternal behavior has been done more than 50 years ago. So we know that. But the interesting part is what specifically is the nature of these neurons? What is the identity of these neurons? And this is a particularly interesting question because the preoptic area does a lot of things. We heard from Peg that it's highly involved in male sexual behavior. It's also involved in thermoregulation, aggression, thirst, feeding. So all sorts of behavior are organized within this area. So it's a mosaic of different cell types. And by candidate approach, we found that the neuropeptide galanin had a very nice colocalization with CFOS positive cells after parenting. It's not 100%, but about 40% of the CFOS positive cells are galanin positive, and about 40% of the galanin positive cells are CFOS positive. So quite a large overlap, not 100% overlap. Moreover, these cells express GABA, so they are inhibitory neurons, and to this day, we still don't know whether their function is provided by GABA or galanin or both of them. But what we've done in subsequent set of experiments is to look at the role of these neurons, first by ablating this population using a galanin-3 mouse and injecting a conditional diphtheria toxin expressing virus. And once these neurons are ablated, we found that now maternal and paternal behavior are abolished, and instead virgin female in particular became infanticidal. When we did the opposite experiment, which is to try to activate these galanin neurons in infanticidal males, we found that optogenetically, we found that this optogenetic activation of MPOA galanin neurons induces paternal behavior in males that are otherwise infanticidal. And so clearly, this neuronal population has the very interesting role of driving parental behavior in both males and females and inhibiting infanticidal behavior. Now, obviously, neurons in the preoptic area tend to have very conserved function. Neurons involved in feeding, in reproduction, have been found to have conserved role across vertebrates. Galanin neurons, would they play a role in other species? We don't know. Recently, both in the poison frog by Lauren O'Connell and in the spawning middleman fish by Andy Bass, in these two species, they found that a strain or species of those animals that were paternal, had increased galanin expression in the preoptic area, and that was the most highly elevated gene when compared males that were parenting and non-parenting. So it is possible that these neurons have similar function in other roles, including in higher vertebrates, but this needs to be studied. So next, we try to have a more mechanistic study of these neurons, which is that, we found this population of, a couple of thousands of neurons, and they organize a really interesting behavior. Interesting because these neurons need to receive input about the presence of pups or other stimuli, about the internal state of the animal, and in turn transform this stimuli into a suite of behavior. Parenting is just not one behavior. It's a whole suite of behavior that includes building a nest, sniffing, detecting the pup, retrieving to the nest, crouching, and lactating. It's also other type of function in the brain that are changed. So for example, the motivation to interact with pups is modified. The sensitivity to hormone, the sensitivity to social cues is very modified in parents compared to virgin animals. And then a lot of other functions are inhibited in parents. For example, infanticide is inhibited, stress is reduced, and interaction with other adults is also reduced. If you wish, when you're a parent, you take care of your pup and you no longer go to the pub with your friends. So you have to make a choice. And your brain is telling you to make that choice. And I'll show you how it does that. So the way we dissected the circuit is using viral genetic strategies such as identifying input based on rabies-based virus. And so the idea is to infect these galanin neurons with a conditional rabies virus that express GFP. These viruses have the ability to jump a synapse and then label the input with GFP. So in a suite, in just one experiment, we can identify all the neuronal input. What are the brain areas sending information to galanin neurons? And what we found was way more than we expected. We found over 20 brain areas sending information to these galanin neurons, way more than what we anticipated. We can guess the function of some brain areas. For example, the nucleus accumbens or the VTA might be involved in the motivation. The periaqueducal gray may be involved in the functional, the motor aspect of parenting. The PVN, the paraventricular nucleus, in some hormonal aspect of parenting. But all these are guesses. Interestingly, when we look specifically at the identity of the input in the paraventricular nucleus, we found that those are mainly neurons expressing vasopressin, and we found no input expressing oxytocin. This is quite interesting because the expectation would be that these neurons are highly sensitive to oxytocin, since oxytocin is thought to be a key hormone for parenting. This is not the case, and I'll show you later when we have molecular data that indeed these neurons do not have oxytocin receptor and have vasopressin receptors. So this is a really nice finding, I think. Then we looked into the projection. How are these galanin neurons fulfilling their multiple function? And so for these, we injected these conditional viruses that enable us to follow their axon projection and synapses using fusion protein between synaptophysin and GFP. And here again, we found a very large number of projections, way more than we initially anticipated. And again, we can estimate that some of these functions would be motor control for the periodical gray, for example, hormonal function for the paraventricular nucleus, etc. But again, these are just guesses at this point. One really important experiment that we did was to try to understand what was the logic of this projection. In other words, is one galanin neuron making projection to multiple sites and therefore sending information and activating multiple other brain function, or is one galanin neuron specialized into controlling one particular function? And so we explored this question by using pairwise dye injection of different colors into two target area, 2 by 2, and then looked at the preoptic area to see whether the galanin neurons were labeled by one dye, meaning they just send one projection, or two dyes, meaning they bifurcate and send projection to those two areas. And interestingly, the results were very clear-cut. We never found, or very rarely found, neurons that were labeled by two colors, suggesting that these galanin neurons do not bifurcate, and that in fact the population of galanin neuron itself can be subdivided into different pools, each pool sending their axon to one particular brain area, and therefore fulfilling probably one particular function. This result was extremely important for us experimentally, because then we can try to study the role of each pool of galanin neuron, the role of each type of projection to a particular area. And so the first brain area that we study is the projection to the paraventricular nucleus. And here the experiment was a genetic experiment, which is that in the lab we have mouse lines that express Cre recombinase under the control of vasopressin, the vasopressin gene, or the CRH gene, or the oxytocin gene. And so what we did was to use, again, this retrograde labeling using Cre-dependent rabies virus. And what we found is that indeed, MPOA galanin neurons project to PVN neurons that express vasopressin, oxytocin, and the corticotropin releasing hormone. So here, related to oxytocin, interestingly, these galanin neurons control oxytocinergic neurons instead of oxytocin controlling galanin neurons. And besides this, also overall have this very important role in controlling the neuromodulation associated with parenting. We then turn to the role of a particular projection, the one to the periacudical gray. The periacudical gray is a very interesting area because it's involved in the motor control of many instinctive functions. And so we thought maybe this is what it does for parenting. So the assay is to put a male or a female together with pups and optogenetically activate the projection to the periacudical gray. So we infect galanin neuron in a galanin Cre mouse with a flexed chondrodopsin virus, and then only stimulate the projection to the PAG, because we know that neurons project only to one brain area. We know we are not activating the neuron projecting to the other brain area. When we do this experiment in female, we found that when the laser is on, the female has more grooming behavior towards the pup, suggesting that indeed stimulation of the PAG projection increased parental behavior in the virgin female. When we do the experiment in virgin male, we found that when the laser is off, most males are non-parental. These are virgin males, so it makes sense. But when we turn off on the laser, we found that now the majority of males are parental. So indeed, this stimulation of the periacudical gray projection increased parental display both in males and in females. We found no change in other social interaction, no change in locomotion. So we think that this particular projection is in charge of one particular role in parenting. That's the motor execution of parenting. We then turn to another fascinating projection. That's the ventral tegmental area, the VTA. And it's interesting because the VTA is thought to be involved in motivation, reward, and so therefore might also participate in increasing parental behavior in the mouse. And so we perform exactly the same experiment as we did with the periacudical gray projection, but to our surprise, we actually found absolutely no effect in parenting, which is that when we stimulate the VTA projection in virgin females, they don't groom more, and when we stimulate the projection in the virgin male, they are still non-parental. So what's going on? And then we start to think about and remind ourselves how much excruciating efforts a mom is able to go through to retrieve her infants. And so this is summarized in this video here. The pup of this mom are here in this beaker. The female just realized that's the case and absolutely wants to reach them. And so she tried to go through the glass. Obviously that doesn't work. Then she realized that, oops, she can smell them or hear them from the top of the beaker. And so she's going to do some acrobatics. in order to retrieve them, and will succeed in retrieving them. So that's what moms do. They will do anything to reach their pups. And so, we tested this, not in that sophisticated assay, but rather just putting a barrier here and just testing whether activating this VTA projection will indeed increase the probability for the animal to go and reach the pups. And indeed, this is exactly what's happening. We found that when the laser is on, the probability of crossing the barrier is way higher than not. Interestingly, maybe the most interesting aspect of this experiment is when we did this in males, in virgin males. So virgin males are infanticidal. We activate this projection. They also cross the barrier with higher frequency. So clearly, the motivation control to reach the pup is driven by the VTA. But interestingly, once the virgin male have crossed the barrier and reached the pups, then they kill them. And so here, remember that we're only dealing with one subset of galanenos, the one that drive the motivation. We're not dealing with the PAG projection, the one that drive the motor control, right? And so here we dissociate in very interesting way the behavior itself to the motivation to reach the pup. And this, by the way, is extremely pup specific. If we put a cookie there, in here, the stimulation of the VTA will not make the animal cross the barrier to reach the cookie. It is pup specific. And then finally, we look at the projection to the medial amygdala. As we've just heard from Peg, the medial amygdala has a very essential role in a number of social behaviors. And so we were very curious to know whether indeed the stimulation of this projection would do anything to parenting or other social behavior. When we looked at parenting, we see absolutely no change, neither in the parenting itself or the motivation to be a parent. But when we now test two adults together, either male-male interaction or male-female interaction, we found indeed that the stimulation of the medial amygdala projection decreased the social interaction. Again, your parent, your galaneons are firing, you stay home watching video and playing game with your kids, and you no longer go to the pub with your buddies. That's what the projection to the medial amygdala does. And so what we've done here is really deconstruct the circuit of parenting, both in males and in female, identifying the various inputs, identifying the logic of projection of these different pools to different brain areas that each fulfill a particular aspect of parenting: the motor aspect, the motivational aspect, the social interaction, and the neuromodulation aspect of parenting. So this is where we were at the circuit level. And these really raised a number of molecular questions or cellular questions related to the preoptic area. The preoptic area, as I mentioned, is functionally and structurally diverse. It's involved in mating, parenting, sleep, feeding, thermoregulation, thirst, etc. And so all of these is confined to this area that has some nuclear subdivision, the function of which is not entirely understood. But certainly the cellular and molecular organization of this cell population is very poorly understood. And so this motivated us means my colleague Zhao Zhang and myself, to try to identify the nature of the cell types, neuronal cell type in the preoptic area, and try to understand their function as well. So associate both the molecular cell type and their function. And for this we use two combined strategies: single cell sequencing of cells of the preoptic area. And I call this actually the preoptic region because, our dissection is not very clean. It's the preoptic area plus the nuclei around, and try to identify the various cell population using single cell sequencing, and then combining this with MERFISH, which is a high throughput in situ hybridization approach that enable to look at the transcriptome of cells in situ. So the advantage of these two strategies is that here this is an unbiased approach. You just look at the gene expressed by individual cell types. You can detect thousands of cells, and this enables then us to identify marker genes that can be used for MERFISH. One problem is that there are two problems with this approach. First, one can detect only highly expressed genes. So essential genes like the oxytocin receptor, for example, that are expressed at extremely low level, cannot be detected using single cell sequencing. And the second problem is that there are absolutely no spatial resolution in single cells, sequencing because you dissociate the cells. And this is a key issue because in an area like the preoptic region, there are multiple nuclei, and you would want to know where are these different cell types located. MERFISH is an in situ approach. It enables to look at the transcriptome in spatially resolved matter. It has extraordinarily high detection efficiency and sensitivity, so one can detect these low expressed, really important functional genes. And it can be combined also with CFOS imaging. In other words, the animal can undergo particular behavior, and now we're going to look at the cell type that express CFOS. And one disadvantage is that at this point, at least, we can only detect the expression of 160 genes, but we actually found this is plenty for what we want. So for those who are not familiar, this is a slide explaining how MERFISH works. It's a strategy that is based on single molecule FISH, means every single transcript is identified in the cell. So every transcript here will be seen, when it hybridizes with a probe, it will be seen as a dot and considered as being on. And if it's not illuminated, but illuminated in the next round of hybridization, this transcript will be 0 and here 1. So the idea is to have several rounds of hybridization with templates that have both a specific targeting region and a readout probe that enable to have a barcoding of the different RNA. And so at the end, if you look here, you go through multiple rounds of hybridization, and you see some dots that are light up or not. And when you combine them all over the various rounds, it gives you a barcode, and the barcode tells you what is the RNA that you've been detecting. And so this we've adapted 2 years ago to brain tissue. This was initially developed in cell culture, and we now are finally have performed our study in the preoptic area and was able to look at the spatial organization of the cells and their involvement in particular behavior. For lack of time, I'm just going to give you a few snippets of our data that I think might be particularly interesting to this audience. One is that one of the particular cluster or cell type that we identified are cell type expressing aromatase. We've heard about these cells. They are absolutely essential for the masculinization of the brain, and they convert testosterone into estrogen. But what we found out of these multiple aromatase positive cells is that, and here they are, so these are the profile of these cells with the various genes that they express, including here a number of interesting hormone receptors, and each of these cluster can be localized in the preoptic area. So here they are, all of them. And interestingly, we found that some of them have, as maybe expected, very high expression of androgen receptor and estrogen receptor. So here one can imagine that now testosterone acts both directly by activating the androgen receptor, by binding to the androgen receptor and activating the corresponding genes, but also by testosterone being converted into estrogen and then acting on the estrogen receptor. But interestingly, so that's this cartoon. But interestingly, some cell type actually did not have any estrogen and sometimes even not androgen receptors, suggesting that there might be a paracrine mechanism such that this testosterone might be converted into estrogen, but not functioning in that particular cell, but rather in a paracrine matter on neighboring cells. And so that then motivated us to look at the location, respective location of neurons expressing the estrogen receptor and not the aromatase. And here are the different estrogen receptors that we found. And as you can see, in this area here, there is a very close proximity between this particular cell type that have aromatase and do not express the estrogen receptor, and then this one, suggesting the possibility of this really interesting paracrine mechanism. We also identify extremely rare cell types. For example, these are the cell types expressing GnRH, so it's a very rare cell type that we robustly identified. Again, we are able to identify the profile of these particular cells, and interestingly found that they have very little or no steroid receptor, estrogen receptor, and androgen receptor, suggesting that they are likely to get their input through other cell types, other input, and in particular through neurons expressing kisspeptin, which is, I guess, the hypothesis, the mechanism that is thought to occur. And when we look now to neurons expressing kisspeptin, we found a very interesting cell type that indeed express kisspeptin among the tyrosine hydroxylase cells. This particular cluster using RNA-seq correspond to a Murphy cluster called I15, which we found here. And this is this particular cluster, which, as you can see, is a very interesting one. It's the kisspeptin of the AVPE. And as you can see, there's a very interesting sexual dimorphism here with way more cells found in female than in male. This, there's nothing new. This was absolutely anticipated. It's well known that there is a sexual dimorphism in cell number in that particular cell type. What was less anticipated is that this is basically the only cell type for which we found a sexual dimorphism. And the reason it's not that there is no sexual dimorphism is that statistically we could not detect them, and we could not detect them because we found an extraordinary high variability among males in the data from males. And this is quite, for me, that was very interesting, because when I talk to my colleagues and they all do behavior on male animal, they always give us an excuse, oh, but females are so variable because of the estrus cycle. Well, in our sample, we had female of various stage of the estrus, and their gene expression was extraordinarily consistent, but males were not. So actually, the variability is in males more than in females, which I think is quite interesting. So Okay, so I'm going to skip the next slide, which is probably not that important, and finish just going back into our galanin neurons. So galanin neurons, as I mentioned to you at the beginning, we think plays this major role in driving parental behavior. But at the level of single cells, actually, we found 11 neuronal types expressing galanin. So it's not one population, it's 11 cell population. And here they are, located all over the preoptic region. And interestingly, what we can do with Murphy is now looking at these cells and look at the ones that are CFOS positive after parenting behavior and identify what are the genes that colocalize with the CFOS positive cells. And what that told us is that there's only one cell type out of the 11, one cell population. That's this cluster here in green called I14, which is found here in green. This one is the only one that is involved in parenting behavior that express galanin. However, we also found other cell population involved in parenting in the preoptic area. I14 is this cluster expressing galanin that is found in both males and females, but we found particular cell type involved in parenting that are only activated in mothers and fathers, some only in mothers, some only in fathers, and then finally some cluster only activated during infanticidal behavior. And so this is, for us, a holy grail where we have been able to identify these cell types with unprecedented cellular and molecular resolution. And I think this is really providing completely new framework for functional analysis of social behavior. So just to wrap up, we identified this, deconstructed this circuit of parenting. We started by thinking about parenting in male and female specific ways, and realized that the circuit both are present in a male and a female brain. And in fact, these bring a more general rule about social behavior circuit that are present both in male and females, but might undergo flexible modulation and therefore provide variation among individuals of a species, as well as variation from species to species, and may also provide fertile ground to try to understand disease. And I want to thank people who participated in this work, and particularly my colleague, Jiaowei Zhuang, who was an equal partner in the Murphy FISH and single-cell cDNA analysis. This was fabulous. We were very complementary to each other. Her as a fabulous technological developer, and me as a neuroscientist interested in social behavior. And then she works at night, I work during the day, so that's fabulous. When we write a paper, I can write, send it to her, and the next morning I have a new version. Fabulous. Thank you.
Well, thank you, Catherine. Well, that was terrific. We're running a little over time, so let's just take a couple of questions now, and we can, I'm sure Catherine will be happy to take more questions during the break. Yeah. Actually, I'm not sure if it's working. You may have to speak loudly.
Okay. Did you carry out your studies in C-June conditional knockout?
In what?
Like C-June conditional knockout? Like
C-June?
C-June.
Why would I do that?
You did your, you carried out on your phospho-C-June, right? The neurons that were positive for...
CFOS, yes.
Yeah, CFOS, sorry. CFOS conditional knockout, did you carry?
Why would I do that?
So that would give you whether it's a cause or an effect.
Well, all we want, the causality is not going to be identifying the CFOS knockout.
Okay.
We want to know the cells involved in a particular behavior. And we do this, we identify this using CFOS. If we want to study causality, we now need to genetically manipulate those populations. But now we have markers, so we can do that.
No, I was thinking for the genetic aspect of it, whether you studied that.
One more question if there is one. No, I think everybody's ready for coffee. As I say, I'm sure Catherine will be happy. So another round of applause. Thank you so much.